A very energetic morning to all my loving students. This is Alpna Shukla, and once again I welcome back to you all in biology class. As we are learning up chapter 6, molecular basis of inheritance. Before that we have learned about the replication. What is replication? Today we are going to learn about the mechanism. So the process of DNA replication take place by the number of substance, enzymes and protein. They are substance deoxyribonucleotides, enzyme DNA helicals, DNA polymers, third, second and first, RNA premise and DNA lyase, protein SSB, single standard binding protein. So as you see the diagram, DNA is the double standard 5 to 3 and 3 to 5 is replicated from the specific point that is called ORI. So mechanism of replication start at the specific point of DNA molecules called point of ORI or ORI, origin of replication. At origin, DNA is standard unwinded by the breaking hydrogen bond. This take place with the help of an enzyme DNA helicals. At the point separation, it appears like a fork or a Y shape. It is called the replication for the K. Each old DNA standard act as the template for the synthesis of new standard. SSB protein attached to unwinded standard and the prevent rejoining. The synthesis of new DNA standard on old standard take place by an enzyme DNA polymers third. It added a new nucleotide through the complementary base pairing. DNA polymers third always synthesize new standard in the 5 to third direction. The synthesis of new standard for the parent template standard having 3 to 5 and is continuous. This standard is called leading standard. The synthesis of new standard for the parent template standard having 5 to 3 and is discontinuous is formed by the small segment of DNA called Okazaki. Fragments, this standard is called the lagging standard. During the formation of Okazaki fragments, RNA premise first the synthesis of RNA primer. DNA polymers third continues the synthesis of Okazaki fragment in the 5 to 3 direction. At the end, RNA primer are replaced by the deoxyribonucleotides with the help of enzyme DNA polymers. The Okazaki fragments join together with the help of enzyme DNA ligase. DNA polymers second once again check and the repair the error occur in the new standard. Now central dogma. What is central dogma? One way flow of information from DNA to mRNA and from mRNA to protein is called central dogma. Mechanism of transcription. The process of transcription takes place by the DNA helicals, RNA polymers second, and ribonucleotides. Ribonucleotides monophosphate activate into the ribonucleosides triphosphate by the phospholysis enzymes and the phosphoric acid. Hence, ATP, ZTP, UTP, and CTP forms means adeno with the triphosphate and phosphate. So transcription being by the uncoiling of DNA at the specific site called the promoter region of cistron. Its place by the DNA helicals, RNA polymers recognize and the bind to the promoter sequence. RNA polymers synthesize RNA always from the 5 to 3 direction. Hence, among two strands of DNA, one standard 3 to 5 and act as the template. You can see the sequence of nitrogen bases or the coding of template standard. The complementary basis of the template standard pair of transcribes mRNA. When the RNA polymers reaches the terminator point, synthesis of RNA stop up. Newly synthesized mRNA is eukaryotic is called the hnRNA, heterogeneous RNA. It undergoes past transcriptional process via the slipping. The eukaryotic mRNA contain the exon and introns. In slipping, introns are removed and the exon are joined in a defined order. hnRNA undergoes additional processing called as the copying and the tailing. In copying in the unusual nucleotides, methyl genericized triphosphate is added to the 5' end of hnRNA. In telling, the adenylate reduce the added of 3' end as a template independently manner. Fully processed hnRNA now called mRNA. It transported out of the nucleus for the translation. That's all for today. Thank you and have a nice day.